Maybe I should, is this better? Okay, I think it's just my volume. Okay, all right. So I have a set of, 3 tutorials. And so what I'll do is just begin by giving some context and just tell you what the 3 are going to be about. And then we'll just begin, right? Okay. So if you look at Okay, so this is a plate from Edward Muybridge, a British photographer, looking at motion, right? And so you have the galloping of a horse here. And what you see is something that looks like, fluid motion can actually be broken up into these discrete elements. And these elements are stereotyped. Okay, there is precise timing at which each of these elements occurs, and they are strung together to create this behavior. And so timing is really critical to many such behaviors, right? Motor behaviors and so on. So one such behavior that relies on precise timing is bird song. Okay, so there are, more than half the species of birds are songbirds. And here is an example of a songbird song. So this is a zebra finch's song. And what you see here is the spectrogram. So along the horizontal axis you have time, along the vertical axis you have frequency. So what you see here so you have these silent intervals and the silent intervals flank this piece, or what you could think of as just an utterance. So that's a syllable. And if you string a few syllables, or the bird strings a few syllables together, so each here is a syllable, and some of these syllables are strung together to form a motif. And this particular bird, the zebra finch, once the zebra finch learns a motif, sings the same motif over and over again. So you see this motif that's repeated. Okay, so each syllable has some distinct identity. The syllables are strung together in sequence, and then these motifs are repeated to form song. Okay. Now this is interesting. So one is it's a sequence that is generated, but it's also a sequence that is learned. So bird song. So many of these birds, or actually the songbirds, all learn their song from a parent, a tutor. Okay. So here is an example of, so this is the adult zebra finch, the father, and that's a juvenile zebra finch. Okay, and this is a learning session, I suppose. Okay, so there's always some discomfort when you're learning new things. Without discomfort, maybe there is no learning. So it's okay. Like I think earlier when Shatavra was asking things and so on, some of us were squirming in our seats, and that's okay, right? So this is a learning session, and there are other birds like zebra finches that also learn their song. Another example is the Bengalese finch. Okay, so the Bengalese finch differs from the zebra finch in that if you look at, now we've sort of transcribed the song, and you can see we have the sequence of letters. It's just a transcription of the song. And the Bengalese finches typically have many more syllables in their vocal repertoire than the zebra finch. There are other birds that have hundreds of syllables in their repertoire. Also, if you notice the song, there is a lot more variability in the Bengalese finch's song than the zebra finch's song. So the zebra finch learns something and then repeats this over and over again. The Bengalese finch is much more of a virtuoso and has all of these, compositions. Okay, so there is interest in understanding how these songs are learned, but also how these songs are generated. So while the acoustics of it comes from, there's neural control, and then there's control of peripheral, bits, right? So like, for example, the syringeal muscles are controlled, and then respiration is controlled in order to create a syllable and then song. Okay. So there is interest in all of these little pieces. So here are some questions that you can ask given this behavior. So one is, how are temporally precise sequences generated neurally? So the control is neural. So how is this temporal precision coming from neural mechanisms? Another question that you can ask is, so what is the syntax underlying these sequences? So I showed you a transcription of the song. And if you just think of, the bird singing thousands of songs, you have thousands of strings, and you can ask, what sort of grammar generated these sequences. And then you can also ask, so these are two different levels of abstraction, right? One is at the neural level. The other is you look at the behavior, and then, you're looking at just transcriptions and asking about stochasticity there. So these are two different levels, and you build models at these two different levels and ask, how do models for the two inform each other? So these are some questions that you can ask, and I hope to address a little bit of this through the 3 tutorials. So the first one, today's, we'll focus on a neural model of birdsong sequence production. In tomorrow's session, we'll look at modeling the sequences. So a particular algorithm, the Baum-Welch algorithm for modeling hidden Markov models, we'll work through that tomorrow. And then in the third tutorial on Monday, just, once you have these models, how do you evaluate these models and say something about, from the statistical models, can you say something about the neural models and so on? Okay, so that's the plan. I am actually more comfortable with these two, that's what I do. But I think the first one, given both the workshop and also to just set context for, whatever else is to come, that's important. So that's what I'll attempt today. But we'll see how that goes. I'm going to pick a simple neural model, and then we'll try and discuss our way through it.
Sumika, I have a question. So, like syntax is a very loaded word, right? Because essentially
natural language syntax.
That's what distinguishes language as we humans understand it from simple animal utterances. So when you say syntax, do you mean that birds have the capability of generating arbitrarily complex utterances?
So one is firstly, semantics is out of the picture in the case of when you're looking at animal vocalizations. The second is that, so we are simply looking at what sort of probabilistic rules exist for transitions between these syllables, and these are typically finite state automata. So we're just looking at what sort of probabilistic finite state automaton can generate the sequence of strings that you see.
Okay.
So that's what I mean when I say syntax.
They have less power than, for example, the kind of grammar that human language typically would
That's
have.
right, yeah. In the Chomsky hierarchy, finite state automata, regular languages and finite state automata. Okay. Okay, so but before we begin, I wanted to say one thing, because it's a mixed group, right? And it was hard to figure out, where to pitch the tutorial. So this is actually, has anybody read this essay called Birds and Frogs by Freeman Dyson? You've read it, okay, all right. So it's a very nice essay, and in it Dyson talks about mathematicians of various kinds, okay? And Dyson says that some mathematicians are like birds, they can survey like huge landscapes and they know how things are connected to each other. And then some mathematicians are like frogs, they know the immediate area really, really well, right? Like the details are really well known. And these are both important perspectives to have, right? So the bird that can survey like huge landscapes, you have the big picture, and then the frog, really familiar with the details. But I also want to encourage you to zoom in and zoom out at your level of comfort. We can be a bit like that, okay? So I will also be endlessly amused that I could look this up, look for this on the internet, and the internet threw this up. So you can be, it's not AI generated. Somebody actually made a frog, I don't know, hat, okay? So we need to be a little bird-like and a little frog-like. So you choose your level of zooming in and zooming out. I've chosen some level of zooming in and zooming out. So some details are missing for the big picture, and then in some cases, like with the Baum-Welch algorithm, there's a lot of detail. So we'll see how that goes.
So did Dyson actually refer Berlin's hedgehog and the fox in this?
I don't think there's a reference to it, but the idea, I guess, is similar, right? But that's
actually it's so similar. I was just wondering, like, with the Dyson.
But that is that like disciplines and.
I mean, he said that like the hedgehog knows only, the fox knows many things, but the hedgehog knows only one thing. Yeah. But of course, he was mostly talking about writers, like Dostoevsky versus.
Yeah, I don't. There's no explicit reference to. Okay. All right. Okay. So to build the neural model that we are interested in, so just a few preliminaries. So firstly, with songbirds, this is known that songbirds have specialized neural circuitry for both the learning and the production of song. And so these areas are well studied. We are interested in this particular pathway. So there is this nucleus called HVC, and then there's this downstream nucleus called RA, which then, downstream is the motor nuclei, which then are responsible for movements of the syringeal folds, etc., for the production of sound. Okay, so this is well known. So we are interested in these areas, and you have neurons. You have functional units that do particular kinds of things. Okay, and we'll use this one. It's a famous experimental observation in the field to think about what sort of neural model we're looking at constructing. Okay, so this is a zebra finch's song. So you see this song motif here. This is, how many of you are familiar with reading this plot? Like what sort of plot is this? This lower one there, and what does it show us? Sorry, spikes. Okay. Just, if you've read this sort of plot before, just raise your hand. Okay. Okay, all right. So then let's just talk about it a bit. So this is a raster plot. Okay, so what you have horizontally is time, and here you have the neuron index. Okay, so these are neurons that are recorded in HVC, the area that I showed you earlier in blue. And what you see here, so if you look at that, so let's look at this inset here, right? So if the rows here are different trials. Okay, so for the same neuron, these are different trials, and each green bar here is when the neuron spikes. Okay, so you have a close burst of spikes, and this is sort of reliable across trials. That's how you would read this plot. Okay, so what you see here, so when they recorded from these HVC RA neurons, meaning the HVC neurons that are from HVC that are projecting to the downstream area called RA. So what they saw was that these neurons, so each neuron spiked reliably at one point in the motif and not any other. Okay, and reliably so. And each of these neurons did precisely that. So each neuron was active at a different point in the motif. Yeah?
Like a sequence of activation.
Precisely, right? Yeah. Singing birds. So in
the first neuron is in the A motif, how will I understand
this one? So
yeah, so you're saying that every neuron in particular neuron in a particular motif.
Part of the, so this entire thing is the motif, particular part of the motif.
Okay.
So this is the, you can see the spectrogram here, and this is just the waveform, the pressure waveform. And you can see, so this entire thing is the motif, and this neuron is active at one point during this motif and no other. Is that okay?
Yeah. How likely is it for a bird to produce a motif again and again? Like for example,
if it's a zebra finch, very likely, because the zebra finch just repeats the same motif over and over again. But a bird like the Bengalese, so these recordings have only been done in the zebra finch. So a bird like the Bengalese finch doesn't do that. So it's the zebra finch's song, you would think of it as deterministic song. Yeah.
How do you differentiate ABC?
How do you know which is what? I mean, so you have to, it's some, well, the processing bit of it is that given the waveform, you actually have to reliably slice the song, and every time the same syllable occurs, you have to reliably identify it as syllable A and then transcribe the song. So there's some pattern recognition at that stage, and then you get the sequence. Yeah. Yeah.
What does it mean that some of the motifs are very compressed? Like green one, red one, but the purple one is very
this one? Yeah, so this is, I mean, I don't know what explains this, but in the neural recording, I mean, so this particular neuron, there seemed to be quite a bit of jitter when it was recorded. It is close enough to that part of the motif, but there is jitter. Yeah. Yeah. So it varies from bird to bird. But typically about 4 to 5 syllables. Yeah, 3 to 5 in fact. In the zebra finch. No, so the zebra finch actually, if the zebra finch learns DBAC, then the zebra finch just goes DBAC, DBAC, DBAC. That's it.
So it's just one name.
Yes, in the case of the zebra finch, yeah. But for example, that's why we would look at something like the Bengalese finch, because there is a lot more variability there in terms of composing. Yeah, yeah.
So why there is gap? Because when we heard the sound, there is no gap, and I mean in our case, I mean we hear the sound as a single sound, but there is a gap.
Yeah, so this is milliseconds, right? So there are silent intervals between the voiced bits. They are milliseconds long. So it just sounds, there are silent intervals between the syllables. Okay, so this was the experimental observation. So these are other neurons in the same area, which are constantly active, but we won't talk about those. Okay. Then there was a different set of, so just to tell you, so this is the motif, and this is a single HVCRA neuron, and it's the same motif that's repeated, right? And you see it spiking, and actually it's a burst of spikes. Okay, at the time it's a burst of spikes. So a second set of experiments was done. This was recording from the downstream area, which is RA. And you can immediately, now that you can read the raster plots, you can immediately see what's going on that's different from what's happening with the HVC neurons, right? So with the RA neurons, you take a single RA neuron, and it is actually active at multiple points within the motif. Okay, so a possible model for how a syllable may be produced, or some acoustic unit may be produced, is that the HVC neurons sort of keep time like a metronome, right? Like going tick, 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 tick. And that's where the timing in the sequence is produced. And maybe each HVC neuron drives multiple RA neurons downstream. These RA, so multiple RA neurons, or rather multiple RA neurons, are driven by the same HVC neuron perhaps. And then the RA neurons drive motor neurons downstream. Okay? And so a combination of this activity is what results in the acoustic output. So this is perhaps a plausible model for what could be happening with these connections across these layers.
So the bounds are coordinated in HVC?
The
source. HVC is how to HVC is only synchronization, or?
So the positive mechanism is that the timing is, the origin is here. And then because of these downstream connections, you then get an acoustic. No, so when you say origin, I guess depend origin of what, right? Yeah. Yeah. So the idea is HVC. There's input from other areas which probably serve as motivation, but then timing is origin. I mean, the hope is that the origin is in HVC. No, but I'm familiar with their work. Yeah. Okay. All right. So we saw a few things, right? So let's think about what are some features of the behavior and some features of the neural recordings that some architecture that you posit should be able to capture. Okay. So as far as the behavior is concerned, so one is that the syllables, there should be temporal stability, right? Which is that once activated, you should have persistence over some time window so that you get the full syllable, right? Second is that the bird is learning these syllables and so on. So previously learned syllables and also motifs and things should be repeated reliably, right? The third is that actually the bird, and here I'm just being general, not necessarily the zebra finch, you should have, I mean the bird can have a large repertoire of syllables. So the architecture should support a large repertoire of syllables, like different kinds of syllables. And then finally, there's also learning. So some birds, for example, can learn new syllables through auditory experience. So the network architecture should also perhaps support learning new patterns. So all of this, it would be interesting to see how all of this comes together in some neural architecture, right? As far as the neural recordings are concerned, we can see that precise activity has to be propagated. And the neurons, whatever the architecture is, should ensure that neurons fire once and only once during a motif. And then that whenever this happens, it's done reliably, sequentially and robustly, right? So all of these features, it would be nice if a model can capture this, yeah. Okay, so the architecture that I'll speak about today is called a SynFire chain, okay? And it's a feed-forward network of neurons. So each is a neuron, each red blob is a neuron here, and there are many layers or pools, and they are sequentially ordered. So this is just representative, like usually there are thousands of neurons in a pool, yeah. And all of these neurons make excitatory connections to the neurons in the next layer. So the idea is that when neurons in this layer are activated, they activate neurons in a downstream layer, and then they activate neurons in a layer going forward, and that's how you have activity propagating down the chain. So there's a signal because of activity propagation down the chain. Oh. Okay. So this, somebody implemented this on Arduino, and I thought you might enjoy watching it. So it's just this idea. What you can see is there's a synchronous volley of spikes from layer to layer, right? And that's how activity propagates down the chain. And sometimes propagation can fail, right? So propagation failure in this context would mean that the syllable doesn't get sung or the motif doesn't get finished.
Different colors in LEDs, do they mean anything?
I don't think they mean anything. Okay, so let's actually try and set up like a simple SynFire chain just to think about what is required for activity propagation along the chain. So so far in the workshop, you've worked with or heard about many different types of like neural models, many that are biophysically realistic. So we're going to just look at a simple what's called a leaky integrate and fire neuron. Taking off, so Shitabro set it up as a neuron as an excitable system which has certain properties, and so we're going to just look at a really simple neural model just to think a little bit about what's required for a SynFire chain for activity propagation. Okay, so we're looking at one neuron, and maybe you can just from what you remember from previous lectures tell me what the form of the equation looks like, etc. Okay, so suppose each neuron, the resting membrane potential is V rest, and this is the differential equation. Okay, without any input that governs the change in voltage. Okay, so yeah, so firstly, can you just quickly maybe, whoever is comfortable with solving differential equations, and you also saw the form earlier, can you just quickly maybe write down what the form of the solution is? If someone can quickly tell me also, that might be fine. So even this morning, actually we saw this form twice today. Both Shitavro and Pranay spoke about this. So what would V of t be? Yeah, okay. So you do all of the integration and everything. And so that would be the form of the voltage, right? That's how the voltage changes with time. So there is no input here, external input. And so what you would see is if the neuron starts at some, so this is the resting potential. And if the neuron starts at some voltage V naught, left to its own, the voltage just decays. So that's just the decay. This is what we saw in the morning. Okay. Now let's say that there is some external input. So we add some current here. And also think about spiking, right? So Shitavro also talked about how, this excitable system, you can think about how beyond a threshold there is spiking activity. So this we just add this term here, okay, which is an external current and a spiking condition. So this is the leaky, integrate and fire neuron. We'll think a little bit about why integrate, why fire and why leaky. The leaky bit is just the decay that we saw. So that's the leaky bit. So yeah, so in general this can be time varying, but we'll first think of what happens if that current is just constant. If you have a constant current input, what does that look like? And also this spiking condition, right? So there's some threshold voltage. If the voltage crosses that threshold, then a spike is emitted. And the condition there is that as soon as a spike happens, the voltage is sort of clamped to V rest. Okay, so if a spike happens, then in the leaky, integrate and fire neuron model, the voltage is clamped to V rest. Okay, so we'll think about this constant synaptic input case. So would you like to take a minute to just sketch what it might look like, what the voltage might look like? There's a constant current input. And yeah.
But this context, in this context,
How is the?
R? What is the R?
You can just think of some resistance such that this is some voltage term.
The resistance would be from the environmental.
Yeah, so biophysically I know, like what could that be? Just the membrane resistance, I suppose, right? Some conductance, yeah, some term that represents resistance, yeah.
Here, there's no, we need a cubic, you use a cubic polynomial to say the firing, to encode the firing.
You don't need it. So let's numerically just sort of see a few examples. So what would this look like? So if you have a constant input, how would the voltage change? Like has anybody sketched it out? Fahad? Okay, so let's take a look at it. So suppose I give you a bunch of, let's say parameters. So there's some membrane constant tau. Okay, so the resting potential, the threshold here, I've just chosen it to be minus 55 millivolts. V naught we start at rest, and there's an input of 2, the current in arbitrary units, an input of 2. Okay, so firstly, will this neuron spike? Janani is saying no. Yeah, why not?
It depends on what the units are.
Sorry?
It depends on what the units of I are.
Say, let's say, I are together in millivolts, let's say, then will it spike? So, and why do you say that?
You need to multiply the current with some time, something with the time dimension which is taught here.
So this is you have a constant current input. It's constant, right?
It will.
So it starts at V rest, and then what happens next? So the threshold voltage here is minus 55 millivolts, right? You start at minus 70 millivolts. And you just have that input there. This here. This is the form. Of course, with, we can consider, because you now have this IR, let me think of V infinity V naught plus IR. Let me just define it this way. So you can again just see what would the solution to that differential equation be. That's just easy, right? You replace. Yeah. Okay. So then what happens when you have constants in equation? If a constant happens.
Shift up. Shift up.
It grows towards what? What does it grow towards? Towards V infinity. Okay, all right. Okay, so that's what it looks like, right? So it grows towards V infinity. The threshold is up there. Okay, so given this setup, what sort of what value would you need for the neuron to spike? At least what value would you need?
I into I.
Okay, so how much given this numerical example? Anything above? You said. Anything above what value would cause it to spike?
40. Minus 40.
40?
Threshold is at minus 50 something.
Yeah, what should this be? What should I change this to such that I will see spiking?
About 25.
Why 25?
I don't know, you inputted like 2 arbitrary units of current, went up to 2 units down.
Okay, so from minus 70 it went to minus 68. And the threshold is at minus 55, right? So
we want V infinity to reach V threshold. We want V infinity to be at or above V threshold.
Okay, so what can I just change this to? Anything above?
Anything above 20. Anything above 25. Yeah, 22.
Okay. So if it hits 55, there'll be a spike, right? Okay. Alright, so I picked 20, but if 15 and above should see a spike, right? Don't need 25, no? Right? Okay. Alright. So what you would see is that the potential rises, and then once it hits 50, so I just use these black dots to represent a spike happening there. Okay, so the voltage just rises, and then here there's a spike. Okay, all right. Okay, so this bit is the integration bit, right? The voltage adds up, and that's the firing. So integrate and fire. And the leaky bit is the decay. Okay, all right. So let's just add elements to this one by one. So now, suppose you also have a refractory period of, let's say, 3 milliseconds. So again, maybe somebody who hadn't heard of these ideas before. So what does that mean, like having a refractory period of 3 milliseconds? In chat. Okay. So what is that? So if once you introduce a refractory. So in 3 milliseconds after it spikes. Okay, all right. So during this absolute refractory period, it cannot spike again. Okay. So for 3 milliseconds, there's no spike again. And so if you had to set this up, so constant current again, but after every spike, for 3 milliseconds, the neuron can't spike again, and then the voltage integrates and it spikes again. Okay, all right. So now let's couple two of these neurons. So we want the Synfire architecture, right, the network architecture. Then I also spoke about coupling neurons and using Hodgkin-Huxley neurons. Let's just think about leaky integrating fire.
Because it's a constant current.
Yeah.
What is the factor that is allowing me to fall back after spiking? I suppose
So computationally, you're just sort of like resetting it. Biophysically, I'm not sure exactly what happens after that. Like I don't know the mechanism.
No, so basically what you're considering is the action potential is so small, action potential usually is so small compared to the refractory period that it's a delta function. So we don't need to actually consider it explicitly. So it just goes up to the threshold, then there's an instantaneous firing which is happening. It's so small that you can just ignore it, and then it's going back to the resting state.
Okay, so suppose you want to couple two of these neurons. Okay. So each is an LIF neuron, a leaky integrate and fire neuron. And this W really represents this RI that we had earlier. Right? And now we think of, so this is the presynaptic neuron. We think of instead of a constant current because the input is coming from that neuron, we think of just a spike. Right? So it's a spike that's passed on to the next neuron, and then we ask, what happens? So then, there's time dependence here, and so if you have that spike, okay. So here, this is where the presynaptic spike comes in at something like 5 millisecond, okay. But the value of, depending on what the value of W is, it can cause. You can cause the postsynaptic neuron to spike or not. In this case, it does not, so that's the threshold again, right? So what happens is that there's a jump in voltage, but there's no spiking that happens, and then the voltage just decays. The leaky mechanism, the voltage just decays back to the resting potential. Okay, so that's what happens here. We can also think about the idea of synaptic delays, right? So, if there is a delay in, when the presynaptic neuron spikes, and then, when the spike reaches the postsynaptic neuron, if there is a delay, then you can imagine that, okay, so these, the change in voltage of the postsynaptic neuron also is, so there are delays that happen here, and this is important because if you look at multiple presynaptic neurons, okay, so the same postsynaptic neuron has multiple inputs from multiple neurons here, then it matters when the spike from each of these neurons reaches the postsynaptic neuron. Okay, so suppose there is some delay, so I've just artificially added some delay from each of these. Then what you will see is, the first spike, so here you can see the spikes come in. So the first spike comes in and the voltage of the neuron goes up, the postsynaptic neuron goes up, doesn't spike, so it begins to decay, and then the second spike comes in, right, and that adds up, and then it begins to decay, and then the third spike comes in, okay. So collectively they push the neuron's voltage towards threshold. And so you can imagine that if you have enough number of these neurons, presynaptic neurons, and depending on what the delay is, etc., you can get the neuron to fire, right? Even if one of them is not able to get the neuron to fire, a collection of them together can get the neuron to fire. Yeah.
Delay, the pulse marks are the time at which the previous neuron fires.
Yeah. Where the previous neuron. Yeah.
So is there any better way to take I not like delta function? Because in general we have seen some weak there, the action potential.
Yeah. So you can use, for example, if you use other neuron models, there are better ways of picking all of these, right? So there are better models. So I just picked this just to illustrate, what are the factors that may matter in propagation of activity in these chains. Okay.
So this
yeah.
marks, this is maybe Akshay question.
Yeah.
So he said that those marks indicate when ABC fires. But is it more appropriate to think of it as ABC are all firing at the same time?
Sorry, delay. Yeah. Sorry, yes. Yeah, you're right. Sorry, that's right. So, because, so that's what I was trying to illustrate with this, right? That, okay, the actual, suppose a neuron fires here. So, we were talking about synaptic delay, right? And if immediately the spike comes to the second neuron, you'll see the voltage change here. If it comes a little later, then you'll see the voltage change here, and it comes a little later, you'll see the voltage change here. So the marks actually represent when the spike reaches D from each of those three. Yeah?
So actually that means that without the delay,
Yeah.
the three would have been sufficient to hit above the threshold.
Yeah.
But because of the delay, it won't be fired.
Yeah. Together they would have, if they had all reached at the same time, would have fired. Yeah. Okay. So so far we looked at a few things that matter. Something else also that would matter is what's called jitter. So this is spike, so variations in spike timing between neurons that are part of the same pool, right? So by same pool, at the moment I'm just thinking of these two, right? So if there is some unreliability in when the neuron fires, this is not the same as synaptic delay. It's not when the spike reaches the next neuron. It's just, the jitter that we saw in the raster plot, for example. Right? Each neuron fires like at a slightly different time. If there is jitter, this will also affect when the spikes arrive at the second neuron and can affect the spiking of the postsynaptic neuron. So this is an exercise. When you have time, you can work on it. So suppose we just look at the small network here and say, there's some jitter that's added to the second input. Okay, so A spikes at t equals 0, and let's say B spikes at some delta t. Okay, so given whatever we've talked about the leaky integrate and fire neuron, can you just figure out for what delta t does the second input arrive too late to push the membrane potential? So there is a small window. If the jitter is within that small window, then the spike will arrive in time to get the second neuron to spike. Okay, but beyond that window, you will have propagation failure. Okay, so can you just figure out what that time should be?
It's the same as lag, right?
Yeah, for a neuron, not when the spike reaches the second neuron. That's what I'm.
If you go to the firing neurons level, it's the different things.
Yeah.
But if you're looking at the perspective of C, they're the same.
Yeah. Okay, so if maybe when you get time, you can just kind of work on this piece here. Okay, just also just a very quick check. So suppose you have just one presynaptic neuron and then there are multiple postsynaptic neurons, okay? And let's say this weight that you need for spiking is 16 millivolts. So do B and C both spike?
The current?
Yeah, it's not like wires where it splits, right? Each of these, it's like a copy goes to each of the neurons. So from the spike from here, a copy goes to each. There's no splitting things. Okay. All right. So where we started with all of this is we wanted a way to keep time reliably for sequence generation. So you could think of, just chaining up these neurons like this. Okay, so this is just a single chain, right? You could think that, okay, perhaps this one is activated and then this one's activated and then so on down the chain. Okay? But this is not very robust architecture. Okay? So any sort of failure somewhere would mean that the activity doesn't propagate at all. Right? And think about everything we said about the behavior, about temporal stability, about repeatability and so on. So you need the network architecture to have all of those features. And so actually chaining them up like this, each pool having multiple neurons, and then passing on activity. So, there is a critical number of neurons that needs to be active in a particular pool for activity to propagate into the second pool. Okay, so some of them may fail, but if you have enough number of them working okay, then activity propagates. Yeah.
Why would they fail? There's no randomness here, right?
So there is, for all of the pieces that we looked at, right, synaptic delay, jitter, it could be the membrane constant of the, I don't know, yeah, the time constant associated with each of the neurons. It could be the starting voltage of each of the neurons. All of these are factors that could affect whether it spikes or not. No?
With a given set of parameters, the behavior, if you do multiple realizations, they would behave the same. They should behave the same way, right?
No, but the conditions for each of them to spike may be different, right? Like they're not.
Oh,
they
they're
need
not
not.
identical.
Yeah, they need not be, right?
Even maybe I'll talk to you afterwards.
Yeah. So I mean, realistically, they need not be identical neurons, right? So in actual, so when in the case of birdsong, for example, when these Simfire chains were made, they were with Hodgkin-Huxley neurons, right? Two compartment Hodgkin-Huxley neurons. So I use a leaky integrating file just to get some of these ideas across. But hopefully, we can all see that this is actually quite nice, right? That robustness is built into this architecture, that you can have a linear chain, but what is nice is, you find a way to separate or to have robust propagation by just having these pools of neurons connecting to each other.
So I guess Akshay's question is like the heterogeneity inherent in the neurons just means that each chain or each neuron is going to behave differently.
Yeah.
But if you give the same stimulus, it will always be identical. The same stimulus.
That's
is
right.
going to generate
That's right.
Exactly the same activity pattern.
That's right. But each neuron could be slightly different in the.
But this means that the same stimulus is always going to generate exactly the same activity pattern.
That's right. So if you look at the pattern of activity that's needed to cause a spike, if you have a particular pattern that causes the neuron to spike and you see it again, it will spike again.
And each row is a replica of, like, are the rows identical or are they also different?
The rows identical in the sense of connections? Or?
No. Is the other rows the copy of first row? You said chain is unstable, not stable, so you added more layers vertically. So are they copies of the first row itself?
They can be, but if you have realistic models, then each of them is slightly different from the one above, right? In terms of structurally all excitatory connections, if you make that choice, yes. But the point being that each of these neurons could be slightly different from each other, and that's the only reason why sometimes there may be failure, right? Yeah.
Adding to that same question.
Yeah.
So when you say the structure gives robustness, is it because of the variability in the inputs?
Because there is variability in the inputs, you need some mechanism built in to account for failure, propagation failure.
So if the input is slightly different, the output would still be.
Yeah, you need it to go all the way through.
This also, in some sense, is capable of giving it robustness with respect to stochasticity. I mean, if you had stochasticity.
Yeah. So if you added some noise.
So if you explicitly have some noise, so that even if you have identical stimulus, each neuronal chain would be slightly different each time. Even then, because of the feed forward and summation, the integration that is happening at each layer from the previous layers, we could actually still give consistent response.
That's right. Yeah.
So that capability is there.
Yeah. Okay, so you see, and so given that if you can have activity propagating reliably down the chain, you can see this kind of firing for different groups of neurons. So just going back to where we started in a minute. So just to show you propagation success and then propagation failure, right? So this is a case where all the neurons in the group fire reliably, and you have a volley of spikes that goes from group to group. Okay, and you can see that with something like this, you can generate sequences, timed sequences. But then you can have propagation failure also, right? So here the spike volley, the inputs are strong, synchronous, well timed, but if there is insufficient weight or some delay mismatches and so on, you will not see propagation happen all the way down the chain. Okay. So for anyone who's interested, an exercise might be, I think Pranay's last class was also saying, look at, or last tutorial was looking at synaptic coupling using Hodgkin-Huxley neurons, right? So you could use a different neuron model and try to see if you can create a synfire chain and have, basically to keep time, right? Can you create a network that keeps time? Use a different neuron model and try to implement it. So just
basically, all it requires is for you to have like a hierarchical layering of neurons.
Yeah.
Each layer has neurons which are giving inputs to the
subsequent layer. Yeah.
And it's only connection from one layer to the successive.
That's right. And no interconnections. Yeah.
And so basically, we should generate like
a set of spikes that go from group to then group and then group. Yeah. And just to go back to where we began all of this, remember the idea was to look at timing that is important to these songs, songbird songs, and to see what sort of neural model can produce that kind of precise timing. So the idea would be this, that each chain, right? So this chain here, this is the architecture we looked at before. One chain here represents a syllable in the bird song. Okay? And so the transitions between these syllables, so one chain propagates activity to the next and then to the next, and that's how you see the motif being formed. Okay. And then the interesting thing is now if we look at the Bengali finches song, which is a stochastic song, suddenly you have the need for these kind of probabilistic transitions, right? So A can be followed by B, but A could also be followed by C. And then if you just look at all of the songs that a bird sings, or well, enough number of songs, you could see that some transitions are more probable than others. Okay, and so you need to set up some sort of competition then between these synfire chains, so that at this point, this branching point, there is a choice to be made between either syllable C being sung or syllable D being sung. So how do you set up competition between synfire chains so that you can account for these kinds of probabilistic transitions? Like that would be a thing to think about. Yeah. So you can ask how can you grow these chains, right? So if you look at the juvenile, just like babies babble, juvenile birds also babble, right? Like they listen to a parent and then they form these proto-syllable like things. They're babbling, and then the babbling at some point crystallizes into song, right? And these syllables are then stereotyped and the motif is stereotyped. So you could ask this question of how do you grow synfire chains? Maybe you can start with like random connections or something, and then just ask how can you grow these synfire chains so that you have certain patterns of activity that persist.
In the case of stochastic songs, would there also be a requirement of, say, between B and C, there are inhibitory connections between them, so that when it takes one path, the other path also doesn't simultaneously form?
Yeah, so in fact that's how we model it, right?
Okay.
So what's typically done is that there are a few different ways to do it. You can have a pool of inhibitory neurons, and that pool makes all-to-all connections to, let's say, the start of these two chains, right? And so when this chain is activated, activates the inhibitory neurons, and that inhibits the second chain. So that's one way of doing it. Another thing is in the Sulfide chain architecture, you can think of a strict architecture where these are all spatially separated and all the neurons are separate, right? But like from a computational perspective, you can build Sulfide chains where actually this neuron could participate in other pools as well. You could think of it as separation in time rather than strict separation in space. So you could use that maybe as a way to think about it.
You don't train these models. Then you have a lot of parameters, like weights and thresholds of each neurons.
Yeah, so if you think of how the chain itself comes to be, like the learning problem that I think Andren was maybe talking about, you'll have to think about an equivalent, right, of learning what the connection.
So you do tune these parameters to.
If you're doing that problem, you will have to.
So to generate these sounds, you need to tune the parameters.
That's right, yeah.
Could you discuss that later, I suppose?
No, so I will stop with Sulfide chains here. Because what I think, can I take like just
okay.
Because what I want to move on to is, so this is the context in which this second set of models that I want to talk about are relevant. Which is, okay, so you have this neural architecture, but you also have this behavior here, right, just the songs. So there's the brain's process, okay, and we call it some cue. And then you get these strings, right, just the songs, the other level of abstraction that we were talking about. And so from there you can build these probabilistic models, some syntax model, okay. And so we want these to be close, like close enough, whatever close enough means, right. So tomorrow I'd like to actually transition to this bit, which is, you know, if you have a bunch of songs, they give you a bunch of transcriptions, how do you build the probabilistic model? So that's what we'll transition to tomorrow. I think that's it for today. Yeah.